Welcome back. Or welcome me back, because I've taken an extended leave of absence of about 2 weeks. I really can't explain it. I felt unmotivated. I was left questioning whether this is all worth it. And then I remembered, I'm not doing it for you guys. I'm doing it for me. I need these review videos more than you need them. Okay, so let's start. Future Kevin. Let's talk about protein synthesis on today's agenda. Now, protein synthesis, what does that mean? That means making a protein. How do you make a protein? You put a bunch of amino acids together in a long chain, you make this protein, you fold it up, and it does a specific function. Okay, but how do you fucking do that? How do you do that? Well, you gotta take your DNA, your genes, and your genes have all the different little instructions for the proteins, and then you gotta express the right gene for that certain protein. Or polypeptide, I should say, because proteins can be made of many polypeptides. So how do you get from the DNA to the polypeptide? I think I heard somebody say it. I think... Oh, transcription and translation involving RNA? That's nuts. Good job, whoever said that in the comments. So, RNA is so much better than DNA. It just sits there, while RNA is boom, boom, boom. It's getting the shit done. You can't do shit without RNA. And there's many different types of RNA. Over the past 2 weeks, I haven't been doing nothing. I actually got a whiteboard and some expo markers, so I don't have to draw on shitty printer paper anymore. But the problem is, I don't know where to hang up this shit. On the wall? Not really sure. But this will help us learn different topics together. So, let's talk about it. You want to make a polypeptide, you say, and then eventually fold it up into a protein. Well, first you're going to start off with the DNA. So say you got a gene, right here. On this one strand, you got a gene right here. How are you going to copy that gene and then eventually turn it into a protein? Well, that's when RNA polymerase comes in, and it does an amazing job. It's great at what it does. So what it does, it comes to the DNA strand, it binds to it, it splits it up within 10 to 20 or 15 nucleotides or whatever. It splits up those nucleotides. Then it uses the template strand as a template and makes complementary base pairs to the template strand. And that's how it starts creating this long mRNA molecule, which is basically the coding strand. Because if you think about it, if RNA polymerase is bound to only one side of the DNA, one strand of the DNA, and it makes the base pairs that are complements of that strand, those base pairs are basically the top strand, except for in RNA, the A's are replaced with U's. It's basically the same thing. So this is the template strand, the thing that it actually attaches to and then complements its bases, and then the top here is the coding strand. Now, how the hell does RNA polymerase know exactly where to go on the DNA strand? Well, it doesn't. That's the thing. It's pretty stupid. So, RNA polymerase requires what's called a promoter on the strand. Say this is the template strand right here. This is only one strand of the DNA, the template strand. It needs to first bind to a promoter, and then once it binds to the promoter, it can then go along the coding region of the template strand and make those complementary base pairs and create that RNA molecule. This right here is a very good visual of what an absolute unit RNA polymerase is. You see, it's doing 3 jobs in one. It's splitting up the helix, it's adding the base pairs, and it's sealing it back up when it's done going over that specific part, and then it chugs along the railroad track. but obviously my picture was better. It represents the new RNA molecule that RNA polymerase has made. This is all happening in the nucleus, by the way. So, okay, you know what's really interesting about this RNA molecule is that there's different segments of it. There's 2 classes, and these classes are called, we'll call them introns and extrons. The introns will be shaded. The extrons will be blank. So these introns, these shaded introns, they're basically the parts of the RNA molecule that will eventually be cut out and thrown in the garbage. And then these exons will come together, they'll come down here, and they'll make the real mRNA molecule. Right now, this is called pre-RNA. It's not yet ready to ship out of the nucleus because it hasn't done this copy and pasting yet. It needs to get rid of those introns and then splice the exons together. Now this is where it gets very interesting because when you cut out these exons, you have different ways of rearranging them. You don't necessarily have to arrange them in one way. You could arrange, if this is 1, 2, and 3, you could go 1, 2, 3, you could go 2, 1, 3, you could go 3, 2, 1. It's all mix and match. You can do whatever you want, man. The cell can mix and match many different combinations with one sequence of RNA. And this is called RNA alternative splicing. This basically means that more polypeptides can be expressed from a single gene. And that's pretty efficient. It's pretty efficient. Then you ask yourself, well, how are these introns being cut out? You know, how are these introns being cut out and the exons being, put together? And it's actually very interesting. These things called spliceosomes in the cell, basically made up of RNA proteins, which we'll talk about. So say this is the mRNA, the pre-RNA strand. Basically, what a spliceosome does is it creates this little bubble. A spliceosome is basically like a group of RNA proteins, and they create this little bubble, and they cut it off. They cut it off. And you know what that bubble is? That's the introns. Those are the introns, and then they paste those 2 exons together. It's really interesting. It's like cutting off a zit. And spliceosomes it's basically a complex of RNA protein molecules, that do this job. They're called ribozymes. And we'll talk about that later. Before the mRNA can actually leave the nucleus and enter the cytoplasm to start translation, because this whole process before that is called transcription. Just want to get that through to you. Transcription. The whole process is called transcription. Jesus Christ. It needs to add a poly-A tail, which is basically a shit ton of adenines, like a shit ton, like a lot, and then a guanine cap. And you might be asking, why does it put on a guanine cap and a poly-A tail? Well, it's because when it enters the cytoplasm, it's a bad neighborhood out there. It's a really bad neighborhood. It's like there's a bunch of digestive enzymes that just want to eat up this, RNA strand and just kill it. So in order to protect itself, it needs to add this and that, the guanine cap and the poly-A tail, to protect itself from the digestive enzymes. Yay! mRNA can finally leave the nucleus and get a job in the real world and find out that life is pretty rough. So mRNA is going to be taken up by ribosomes in the cytoplasm, bound or free, and those ribosomes are going to make the protein using the mRNA as like a construction booklet in a Lego set, So, well, then you come across this dilemma. If ribosomes make proteins, and proteins make everything else in your body, how the hell did your body make ribosomes? right? Like how? And the answer is actually very cool. So if you look at the anatomy of an RNA molecule, RNA molecules, they're made of amino acids in them with, phosphate groups and ribose sugars. But they have amino acids in them. And what is a polypeptide? Well, a polypeptide is a long chain of amino acids. So you see some similarities between the two. So within RNA, those amino acids can actually bond to each other. They can fold on top of each other, and they can create that 3D structure that a protein has. And that's what a ribosome is. A ribosome is made up of RNA. Ribosome, RNA, RR. It's really cool. Same thing with the spliceosomes that I mentioned earlier. Those are basically RNA molecules as well. And they fold on top of each other and they make that shit. And we'll talk about tRNA, which is another form of RNA that folds onto itself to act like a protein. It's really cool. Within a ribosome, you have the large subunit and the small subunit. And they come together and they take in that mRNA molecule and they basically start the translation phase. Translate. They translate the RNA into a sequence of amino acids. Within the RNA, there's a bunch of base pairs. Every 3 base pairs is called a codon. A codon. Every 3 base pairs. Triplets. And each codon codes for an amino acid. Different codons can code for the same amino acid. There's some redundancy there. Because like in a codon, say you have AAG and AAC. The cell doesn't fucking care about that last base pair. It's already got the first 2. If the first 2 are the same, generally it's going to code for the same amino acid. That's why the 3rd base pair of a codon is generally called the wobble, because it doesn't fucking matter. So let's draw a picture of a ribosome, shall we? So here's our ribosome right here, the large Krabby Patty looking buns, looking fat buns. Large subunit, small subunit, and then the mRNA is lodged in between them with its nucleotide strand on it. So how the hell is this ribosome gonna turn this sequence of nucleotides into a sequence of amino acids? Well, it needs a little help. It does. And it gets help from molecules called tRNA. tRNA molecules. Another form of RNA. What's a tRNA molecule? Well, it's in the name basically. It's another form of RNA folding onto itself and creating like a protein type thing that does a function within the cell. And its function is to bring in free amino acids in the cytoplasm. Say this is amino acid in the cytoplasm. The tRNA will bind to that amino acid with the help of an enzyme called amino acetyl synthetase. You don't need to know that. I just sounded really smart saying it. It's building my ethos, my credibility. The anatomy of a tRNA molecule, as I said earlier, it's an RNA molecule that's been folded onto itself and creates this 3D structure. But also at the bottom of it, it's what's called an anticodon. It's the anti to the codon on the mRNA molecule. It's basically a complementary pair of 3 bases that reflects the mRNA. That's how it binds to that specific part. And that's why a specific codon will code for a specific amino acid. So within the ribosome, you have these 3 sites, A, P, E. The A site is where a tRNA molecule comes in, the accommodation site. The P site is where the growing polypeptide chain lives on the middle tier tRNA molecule. I'll explain this more thoroughly. Bear with me. And the E site is where it exits. At any given time within the ribosome, there is 3 tRNA molecules. I'm not just showing one going to the next step to the next. There's 3, and it's moving down like a chain. Gets rid of this one, gets a new one. Gets rid of this one, gets a new one. And every time it gets rid of that one, all of them shift over, and this like chain, the amino acids will covalently bond together, and this one that was originally in the A site will move to the P site, and the one that was first in the P site will move to the E and get kicked out. And then another tRNA will come in and take the place of the old one that was in the A site. I know, kind of confusing, but I explained it like a G. You're welcome. That actually got me sweating explaining that, because it's quite a challenge to explain something this like intricate, I guess, using a shitty whiteboard and a couple of markers. It'd be really cool if I could put in some animations, but we don't have the budget for that. Maybe if you told some of your friends and your family about the unprofessional tutors, TUT, might get some more funds going in here. Because currently I'm making $0 to do this shit. So, you're welcome. Anyway, so once the ribosome finishes doing its job, cycling through the tRNA molecules and having its long chain, you got your polypeptide now. And sometimes when a ribosome is first creating that chain, the first few like things that it creates, the first few things of that polypeptide that it makes, it will actually serve as like a signal for the cell to take that specific ribosome and its polypeptide and bind it to the ER. Remember, there are things called bound ribosomes that specifically make proteins that are destined to be shipped out of the cell. And those ribosomes that are flagged for secretion, they will bind to the ER, make their protein, and that protein will go through the ER and then go through the Golgi, and then it'll get shipped out to do like 3 tours in Iraq. I'm trying to think of other things to say, but there's really nothing. I basically like summed up years of research in, I don't know, 15-minute video. I think I did pretty good. And hopefully I'll have the willpower to create another episode today. Yeah. Also, fun fact, when it goes through the when the polypeptide is done being a long chain, things called chaperonins. It's basically like a hydro flask, and then you shove the polypeptide in there, you shake it up, open the lid, out comes a regularly folded protein. Don't know how it works, that's just the way it is. That's a wrap. Later, bitch!